haven't met, I'm Stacy Kalish. I'm a longtime board member and currently the vice president of the board. I'm a really long-time NTSAD member and the child of a Tay-Sachs carrier couple. So my parents found out that they were carriers of Tay-Sachs when they were first married, before they had their children. So our goal this morning is to talk about a couple of topics in research. So I'm going to talk briefly about NTSAD's research efforts, just so that you have an idea of what we try to do and how we try to do it. We're going to hear a keynote speak from Dr. Walkley. We're going to talk a little bit about how families can become involved directly in research, really even now, in us learning about these diseases. And then we'll talk a little bit about the road to clinical trials. And then we're going to break up into small groups based on disease type, and you'll get to really sit down with some of the people who are doing this work and talk to them directly and ask them questions. This whole session is really designed for you guys, so please feel free to ask questions. If you don't understand things, ask questions, especially in the small groups. We've asked our speakers to really speak for short periods of time because we really want you to be able to have conversations with the researchers. So the NTSAD Research Initiative was founded really in 2002, so we're going on 16 years. And the goal was really to help accelerate the path of research and accelerate the road to treatments and to cures by collaborating and bringing together the clinicians, the scientists, really the families and the patient organizations. So in the 15 years of granting, NTSAD has now funded 61 projects from NTSAD alone, and then an additional 13 projects when we were working with the Lysosomal Storage Disease Research Consortium. So these are a lot of projects that we funded, and we'll talk a little bit about what those look like. And NTSAD has granted almost $4 million worth of money in the last 15 years. And the real goal and the strength in this money is that it's gone on to allow these researchers to receive larger grants from organizations with bigger pockets, and has been leveraged to receive over $21 million of additional funding based on information that's come out of these grants. And the process that we use to choose what we fund is really based on NTSAD's research strategy. So our strategy really focuses in 3 different arms. The initial focus of the research initiative early on was really to fund these seed grants, which is looking at early stage research to provide small grants to allow researchers to collect the data that they need so that they can then have the information needed for larger grants. Over time, we've really moved and have supported quite a bit of work in the gene therapy realm, which you'll hear much about later. And then we also work to try to support things that are needed to get us towards clinical trials. So what do these look like? The initial seed grants can really take a variety of shapes, and these really look at novel approaches, so either novel therapies, new drugs, or novel approaches and new ways of delivering medications. So some of these were small molecule medications. These are new medications that may help the body store less material or may help any remaining enzyme work better. We've supported projects, again, that look at new ways of delivering therapies, including a therapy that we supported that studied giving enzyme replacement through an intranasal or through the nose approach. And then also we have supported projects that do really important work of developing cell and animal models. So these are either cell lines or animals that show signs of these diseases so that researchers can study whether or not their potential therapies are effective. You guys will hear plenty about gene therapy throughout the course of the morning, but we've supported gene therapy programs and work in gene therapy programs really for all of our diseases. And also this includes work with a number of different approaches. So you'll hear about some approaches to deliver gene therapies directly into the brain and others that may use IV approaches. And then also some combination therapies that look at combining stem cells and gene therapies. And if you're not really familiar with what these types of therapies are, you'll hear about these in your small groups from the people who are doing this work. And then finally, a large part of our discussion has been around clinical trial readiness. And this is really sort of the foundation that's needed to build the house of your clinical trial. So we'll talk more about clinical trials later, but the goal is really to know if an experimental therapy is helpful. And to know if something's helpful, we need to know what the disease looks like before the therapy. And so these clinical trial supports take those forms of biomarker studies, so looking for markers in blood or spinal fluid or imaging studies to see what the natural course of the disease is. It includes natural history studies, which includes collecting information from families and from their physicians to find out what the developmental milestones are and what the other milestones of diseases are. Some of the wearable technology projects that many of you have participated in, also talking about newborn screening and better understanding of our genetic testing technologies. So the way that we choose what we fund is through a request for proposal or RFP process. And so we solicit, just to make sure that's what we're, so we solicit projects really for any of our diseases, and these projects can be anywhere along the spectrum, from early novel therapy basic research all the way up through studies that are needed to support clinical trials. And so we use a two-part process. So we put out a request for proposal that anyone can submit a proposal to. It's a shortish one-page document. And then our scientific advisory committee, some of our other advisors, help us select from that larger group which proposals seem most promising, and then we request those researchers to submit a full application. We again work with our advisors to then make choices, and the way we choose what we're funding is really based on the scientific merit of the project, obviously, also how feasible it seems, so if the project can be done in the amount of time that's proposed and for the amount of money that's requested, how likely this is to be successful, and also where there's unmet need. And this is just a picture that sort of shows where some of the things that we talked about are in this process. So again, we support work from the beginning of discovering new drugs and new approaches through the models and early work that's needed to show if these may help into the support that's needed for clinical trials. And so this, again, is the plan for the rest of the morning. So unless there's some questions right now, I'm going to turn this over to Dr. Walkley, and then we will come back later. Questions?
Most of you don't know me. I don't know most of you. I've been at this meeting a couple of times, and I really appreciate Sue's invitation to come and be with you this year. I'm a basic scientist. I've been working on lysosomal diseases all of my career, which is almost at 4 decades now, and done a lot of work on Tay-Sachs and Sandhoff disease, particularly earlier in my career. But the last numerous years, I've been very involved with another lysosomal disease called Niemann-Pick type C, which I'll say a little bit about here. But what Sue has asked me to do is just come and give you my sort of sense of what's going on in the field of neurometabolic disease, particularly with lysosomal diseases like Tay-Sachs and the others, as well as Canavan disease, and then talk a little bit about the future of and where we're headed. So there are going to be 3 themes here. First, I want to, in very basic lay language that I hope I don't insult anyone's knowledge here. I realize in an audience of this sort, some of you have been involved in this challenge for years, and others of you are newer. But I want to talk to you about some of the excitement going on in the field, particularly around the area of lysosomal disease, and then try to make the argument to you that understanding these diseases is really, really critical, that we can't just go after therapy, we have to also go after our understanding of how the disease is really affecting the brain in particular. And then finally, with Sue's encouragement, I want to talk about a collaborative approach to developing therapy for this disease, Niemann-Pick type C, that I mentioned, and how parents were involved and some of the things that happened in that experience. I think you might find it interesting. So let's start with a cell. Start with a very basic view. And is there a pointer? Yeah. Oh, I see. Excellent. Okay. So slow motion pointer. So our bodies are made up of cells, and our cells are marvelous little machines. It's just, I never cease to be astonished by the beauty and complexity of ourselves and our bodies and the way cells work and the way thousands of proteins are interacting every single moment of the day and going about their business and just the marvelous nature of this. And of course, our key interest here, I'm going to start with lysosomal disease. We'll talk about Canavan in a few minutes as well. But let's start with lysosomal disease. And here's the lysosome, this little organelle in the cell. It's sitting here, in the cytoplasm of the cell. There's a nucleus where the genes are and a endoplasmic reticulum and Golgi apparatus where genes are, where proteins are made and carbohydrates attached, and they are trafficked out into the cytoplasm to other organelles as need be. Of course, there are mitochondria in here as well, the little energy sources for the cell. But the lysosome, it's sort of a when people talk about cells, sometimes it's the last organelle they would mention. It was discovered only in the 1960s, and it's I think it's fair to say up until very recently, it's sort of been looked at as sort of a little cul-de-sac in the cell, a digestive system, and I'll say a little bit more about that in a minute. But here it is, and within a cell, there would not just be one lysosome, there would be many, and they would be out in the cytoplasm. And we can take a diagram, actually take it out of a science paper. This is from Paul Saftig, very prominent scientist in Germany. And here's that same little organelle. Now it's drawn out. It's got all these little squiggly things, which are proteins in the membrane of that lysosome. Just giving you a little bit of a sense of, it looks so simple here. It still looks pretty simple here, and it's even more complex than this. But that membrane contains all sorts of proteins that are critical for different aspects of the lysosome to function. And in addition to that, there are also, within that lysosome, lots of other soluble proteins that are enzymes. And of course, many of you already appreciate that the lysosome has this digestive function for the cell. And I'll say again, it's a recycling center, digestive system for the cell. Go back over that in a little bit more detail in a few minutes. But these enzymes within the lysosome, which, by the way, has a slightly acidic environment, and the enzymes that are present in here work best at this acidic environment. This is, in fact, how the lysosome and its enzymes was initially discovered, was because of that difference. But in the last, it started about 10 years ago, and then about 5 years ago, some other discoveries happened. And I would say right now, we are looking at a whole new vision of what the lysosome is doing in cells. And in fact, looking back over my career and thinking about the discovery of the lysosome, I would say we're in an era now where the findings that are coming out about the lysosome are equivalent to that of its actual discovery back in the 1960s. It's that exciting. And it comes from the finding that there are some other proteins associated with the surface of the lysosome here. I'm just going to simplify, let's just call it the regulator complex, that has an association with another protein, which I'm going to call mTORC1. It actually stands for the mammalian target of rapamycin complex 1, but we'll call it mTORC1. So another protein can either be in the cytoplasm out here, or it can be attached to this regulator complex. And then there's one other player that's been discovered that we'll call TFEB, which is a type of, the TF stands for transcription factor EB, but we'll call it TFEB, which again can be in close association with this mTOR. And what we've learned that's so exciting about the lysosome is that this mTOR1 TFEB signaling axis, we'll call it, it is actually acting as a nutrient sensor for the cell. And what do I mean by that? So the lysosome is this digestive organelle, if you like. And so there are complex molecules being brought into the lysosome. They're being broken down by these enzymes. And then there are precursor compounds, smaller molecules being generated by that digestive process that then have to go across this membrane. They have to egress, they have to leave the lysosome. And they do it sometimes through these proteins that are shown here in that transmembrane space. And so the lysosome is this recycling center where complex molecules are coming in, simple molecules are going out. But we've known that for a while. But what we know now is through this signaling axis of mTORC1 and TFEB, there is this mechanism whereby the cell is able to regulate its overall nutritional state. And I'm going to go through that in a little bit more detail, but that's the critical take-home message. And ultimately, this process with TFEB in particular is not only regulating that nutritional state, but it's doing it in part by controlling how many lysosomes there are in the cell, and also whether a process called autophagy is being turned on. Now, if you like to break down the meaning of words, this one is actually pretty easy. Autophagy, auto, self, phagy, eating. So autophagy is basically self-eating, and what it is a process whereby the cell will break down organelles and recycle them. And this is another part of the beauty of our cells. Every molecule that we synthesize gets recycled at some point. That's why, again, why the lysosome is so important. Everything is being recycled all the time. And one of the major ways that this material gets into the lysosome is actually through autophagy, where a bit of membrane surrounds like an old mitochondrion and engulfs it and forms something called an autophagosome, which then goes over, fuses with the lysosome, and then breaks down these proteins and other materials that are inside that old, worn-out mitochondrion. So that's called autophagy, and this mTOR-TFEB process is regulating that the extent to which autophagy is occurring. So in other words, if a cell is running low on nutrients, it can rev up its autophagy and thereby generate more of that, the needed small molecules for all the various metabolic pathways that are active in that cell. So here's a little bit more complex diagram, but it's really still, hopefully it's not too complicated. But I just want to stress this idea of a high nutrition state versus a starvation state. And let's start here on the right-hand side of the diagram. And so this is actually the gray area here is inside that lysosomal lumen. So you can imagine this is the membrane of the lysosome going across here, and sort of just imagine that as a big circle that's going around down towards the floor. And so here we're inside the lysosome, and let's say we're in a state where there is lots of nutritional material around. We say high amino acid content. Amino acids, of course, being the building blocks of proteins. In a state like this mTORC1 protein is anchored to this regulator complex. And when it's there doing that, it can carry out its normal function in the cell, which is it's a kinase. So it's adding phosphates, it's phosphorylating proteins, which can activate proteins or deactivate proteins. And you probably can't read all the small print in here, but the purpose of this diagram is just to tell you when mTOR is active and it's phosphorylating proteins, it's driving all sorts of growth processes within the cell. It's actually, mTORC1 is really of great interest to cancer biologists because of this feature that it has to do with cell growth. And it can also be involved in cancerous cell growth when it's out of control. But But so this is a state of high nutrition, and there is this cell growth that's part of that.
Excuse me, Steve.
Yeah.
Can I ask a question?
Sure.
This is a little too complex. For me, I was wondering if you could, you're talking about nutrition. Is there something connected to the diseases and what the implications
Yeah,
are?
I'm headed that way.
Yeah, because this is, I think, I'm not a scientist, so I'm kind of just talking on behalf of others.
Okay.
If maybe, if
This is the most complex slide,
Oh, good.
so
So
I
maybe
promise
we
you.
could move on to that because
And will you make this, can people get this after if they want,
Sure.
they want this level
Sure.
of detail? As
Sure.
you're hearing this
Okay,
level of
so
detail
you're
right
telling
now?
me I need to move on.
I'm just trying to say, what is it?
I guess a question is, does what our kids eat make a difference? Because this is a nutritional thing.
Yeah. We could talk about that later, I'm afraid, if I go into that. I mean, there is a link,
Okay.
but that it would be too complicated. Let me just try to fit, just for those of you who are following, and I apologize for so one of the things that happens when this mTOR complex is sitting here and it's very active is it's also phosphorylating this other player, TFEB. And when it does that, it keeps TFEB in the cytoplasm of the cell. So it can't be active. TFEB is a transcription factor. It has to get into the nucleus of a cell in order to be active. If it's got this phosphate group on it, it can't get in there. And then what happens in a state where there's low nutrient availability? And what happens there is that the mTOR complex 1 dissociates from the regulator, it's not active. And so TFEB is not being phosphorylated, it can go into the nucleus. What it does when it's in this non-phosphorylated state, it can go into the nucleus and it enhances genes that lead to the expression of all sorts of lysosomal enzymes. It increases the number of lysosomes, and it also enhances autophagy. So, in all of this, what I'm trying to say is that the lysosome has become this really remarkable regulator of cell metabolism. And we've never been in this space before. It's always been, this lysosome, the last organelle in the cell that people would consider important and so forth. And of course, all of this, for those of us working on lysosomal diseases, now to have the lysosome in this really pivotal place of when this mTOR1 is sitting on the surface of a lysosome, it's really regulating the metabolism of the cell. It completely changes the story. And so when we think about a lysosomal disease and what you're looking at here is a neuron's actually in a ganglioside storage disease, GM1 gangliosidosis, and it's completely filled with the storage of this GM1 ganglioside. We have a brand new handle on beginning to understand why that storage is actually causing brain cells to malfunction. So that's really the take-home message from that. So we've gone over all of these many years since the discovery of the lysosome. In the beginning, it was thought of as a suicide organelle, and then a digestive system, and then a recycling system. But all of this it's still sort of a cul-de-sac, just an end organelle, and a lot of people didn't find it all that exciting. Today, we can think about this greater lysosomal system, and we can really put the lysosome into this sort of much more central role in the cell itself. And I like to call it the center of the cellular universe. I think it's really a master regulator of what goes on in the cell in a way that we've just never appreciated before. And so, those of us who are studying these diseases, we really appreciate how complex the neuropathology is in the disease. And, we've got storage, which we know a lot more about now and how complex it is. We've got other changes to cells, including their dendrites and axons in terms of neurons. There are other features in the cells that have nothing to do with the lysosome. And we've never really been able to get a complete handle on why all of these things are happening. And so just to, look at it again in this sort of cartoon fashion, lysosomal diseases ultimately are very complex. It all starts with a single gene mutation, loss of a single protein, but then this huge cascade of events takes place. And what we're trying to do is work out, what happens next and next and next and what causes what through this series of events. And of course, we know that neurons can live with these diseases sometimes for many years, but they begin to malfunction. And so our goal is to understand all of that. And this new discovery that I tried my best to explain to you, and I apologize if I was a little overwhelming on it, is really giving us a new way to look at the cells. And then, my second point here is that these insights end up helping us come up with new ways to devise therapies. And, we know in terms of the lysosomal disease area, we know that enzyme replacement therapy has been around for a long time, very successful in some types of lysosomal diseases, particularly those without brain involvement. Getting that enzyme into brain has been difficult, so it's been limited. There are other approaches like substrate reduction therapy, which again has worked well in only a limited number of lysosomal diseases. Niemann-Pick type C that I work on is one of those, not so much in Tay-Sachs and so forth. Other categories, chaperone therapy, proteostatic regulators that attempt to get a defective enzyme in the cell to the lysosome where it could function. Those studies are underway. But in terms of what I've just been talking about, another whole approach that certainly your research committee will be considering, I'm sure, involves this area of drugs that manipulate this mTOR TFEB signaling axis, because this is one of the hottest new areas where there is some data emerging in some lysosomal diseases that argues that it could be beneficial. And then, of course, there's the exciting area of gene therapy that Doug Martin and others will be talking about today. And again, a very important area in its own right. But let me leave lysosomal disease now and just speak for a minute about Canavan. I don't work on Canavan disease. I've sort of followed it very from a distance over the years. Of course, it's not a lysosomal disease, but it's a disorder that has intrigued investigators for a long, long time. Many therapy approaches have been tried. It's a condition where there is this brain metabolite, call it NAA, N-acetyl acetate, NAA, one of the most common molecules in the brain. Very, very abundant. We know it's synthesized by neurons. We know it's taken up by cells called oligodendric glia, which are the cells that make myelin, which is this fatty substance that's critical for, you can see it's along the axon here. It's critical for axons to be able to conduct their action potentials, their way of signaling to other cells. And what happens in Canavan disease is there is an absence of this enzyme, aspartyl acylase, ASPA, which can only be found in oligodendric glia, and which normally breaks down this NAA into free acetate, which is then available for this other synthetic process that's critical for the formation of myelin. And there have been all of these, over the years, questions about the, again, the pathogenesis of this disease and whether too much NAA was responsible and it's toxic, could be killing cells, versus not enough of this acetate within the oligodendroglia to allow for the synthesis of myelin. And this disease is largely a disorder of too little myelin formation. And so when we look at therapy approaches, again, replacing enzyme replacement therapy, replacing this enzyme, I don't think, it's been tried, but it's not been successful. Replacing the missing acetate in the oligodendroglia, it's kind of interesting. I was not aware of this, but looking at clinicaltrials.gov, there are clinical trials going on to look at acetate treatment to replace the low level of acetate in oligodendroglia. I'm not sure where the status of that trial is, but it's ongoing. And then, of course, the other question is, could you do gene therapy here to replace that enzyme? And again, not an expert in this area. Others of you here in the room know far more about it than I do. But I was interested to see, just in looking at what's in the literature that's new, this paper that just came out in the early part of this year in journal Acta Neuropathologica, where complex paper, a lot of different findings reported in it, but the two that really caught my eye is they made 2 mouse models as part of this paper. One, they overexpressed NAA through a genetic means, but it didn't create a model of Canavan disease, really arguing that it's not just too much NAA that's neurotoxic that causes this disease. The other model they made is they deleted the ASPA enzyme just in these oligodendroglial cells, and they successfully created a model of Canavan disease, suggesting it really is this oligodendroglial component and the lack of the enzyme there that's most critical. And then they did, interestingly, with I believe an AAV2 vector, they did gene therapy in the second model, and they were able to correct the disease phenotype. So progress is being made here with Canavan disease as well. So with that said, I want to spend the sort of last little part of my talk, if I have a few more minutes, I do. I want to sort of shift maybe something a little less difficult to understand. As I mentioned, I work on this disease, Niemann-Pick type C, and I got involved in some collaborative work now almost 10 years ago that involves some of the players that are and institutions that are shown here, including Dan Orey at Washington University School of Medicine, Charles Feet at the Children's Hospital in Philadelphia, and myself, and then Denny Porter, a physician scientist at NIH. And this collaboration, as you'll hear as I go through this, is supported by a really unusual means, but it's called SOAR, which stands for Support of Accelerated Research for Niemann-Pick C disease. And this story started back in 2007. I'd gotten to know a father of a child with Niemann-Pick C disease. We've become actually very good friends. And he heard about a very wealthy banker who lived in Brazil, who was retired. But he had a daughter, the banker had a daughter with Niemann-Pick C disease. And my friend thought, let's go visit this person and explain to him the need for funds to support research towards therapy for Niemann-Pick C disease. And maybe he would be generous and support that effort. And as part of that, the father, my friend, and I, along with Mark Patterson, who some of you may have heard of, very prominent pediatric neurologist, and Dan Orey, who I just mentioned, the 4 of us went down to Petrópolis, just outside of Rio de Janeiro in Brazil, and we spent a long weekend on the estate of this wealthy banker. And it was really an amazing experience. we had a colleague from Brazil come in and translate. We presented scientific talks to him. I can see now I may have overwhelmed him because I think I gave a talk even more complicated than what I did just now. And maybe that's the explanation for why, after we did all of this, he didn't provide any money at all to research. But what was important was on our way down there, we thought, well, if we're gonna ask this wealthy guy for money to support research specifically towards therapy for Niemann-Pick disease, we need to have a plan. We need to have some kind of organization, and so that's where we created this entity called Supportive Accelerated Research for Niemann-Pick disease, and called it SOAR. Went through a lot of other eponyms on the way to getting to SOAR, but that's the name. Mark Patterson actually came up with it. And then, so we got back, and he didn't give us any money, but we kept this idea going. And again, it was partly through the sheer energy of this father that is here. And so our first formal meeting, and you can see it was a sizable meeting, was actually in a classroom at Stanford that we were able to borrow. Scientists, physicians, families, caregivers, other interested parties, we all gathered together here and to talk about, and Denny Porter is here, Mark Patterson is here, Dan Orey is here, Liz Neufeld, some of you may know, is here, other scientists, quite a few families. And we really talked about how we could move an organization like this forward. And in the end, we created this, and I'm going to show you that in another slide in a minute. But the whole idea, and of course it was just crazy idea, was we were going to develop a therapy to present to the FDA for Niemann-Pick disease within 3 years. This, of course, is the father speaking, the scientists. We knew this was a hill too high. Am I running over in time? Should I wrap it up? Okay. I thought you were signaling me to. I'm almost done, actually. Okay. Gustavo and I were talking about this over dinner last night. Treating genetic brain diseases, everyone in this room knows this, it's exceedingly difficult. And Niemann-Pick disease is a very difficult lysosomal disease. I commented to this father, in fact, early on, that it was probably because it's actually a defect not in an enzyme in the lysosome, but in one of those transmembrane proteins. I commented to him that it'll probably be the last lysosomal disease treated. But you'll see, I was wrong. So we created this organization, and just to sort of walk you through the guts of it, it consists of a core of scientists. Again, it's myself, Charles Veet, Dan Orey, and then we have a project manager who is a former graduate student of mine, who is an absolutely essential part of keeping us organized, because we have conference calls every 2 weeks where we plan experiments together, all directed at therapy. We test different agents in our mouse models, and critically, one of the beauties about the system that we have here is Charles has a naturally occurring cat model of Niemann-Pick disease, and this group would be very familiar with the value of large animal models of lysosomal diseases. And so there is a feline model of Niemann-Pick that is at the University of Pennsylvania. So that's the core group, and we collaborate very closely with the families that support this organization. That group has grown from 1 to 2 to now over a half dozen significant contributors. We have face-to-face meetings at least twice a year. We have regular conference calls, and so there's a great deal of interaction between the scientists and the families. And again, part of that is coordinated by our project manager, but we also do that with them. We also have close ties to NIH. NIH is where the natural history study is occurring for Niemann-Pick disease, and where the clinical trials have taken place, including the one I'm going to tell you about in just a minute. So that's a very, very critical player. We also have a scientific advisory committee, which particularly early on, these were senior scientists that weren't part of the actual research, but they were here to sort of advise on our progress and our plans within the scientific collaborative. We use them a little bit less now that the organization's more mature, but they're still part of our plan. And then we also have the ability, when there are projects that we want to pursue that's outside of our capabilities, we have the ability to work with the families and with particular labs and develop other sort of side projects where these scientists then report back to us and then we report back to the family. So we're able to, in a very flexible way, to expand our expertise by recruiting in additional scientific collaborators. And then finally, of course, all of this sits within the larger Niemann-Pick C community and lysosomal disease community. We have close interactions with some of the major players, the Ara Parsegan Foundation and the National Niemann-Pick Disease Foundation, as they work with the Niemann-Pick C community as well. So, sorry? Okay. All right. So, so the positive thing that happened, and I'll just go through this here, is that a therapy was discovered just about the time that that we were starting this NPC-SOR mechanism, and it came out of my lab and out of John Dietschy's lab at UT Southwestern, and it was a drug called cyclodextrin, which some of you probably have heard of, that ended up being very effective in Niemann-Pick C disease. And it was really through that discovery, and I was going to walk you through the timeline of this, where we start out with the initial discovery of cyclodextrin, then working with NIH, and then NIH bringing in a biotech partner. This drug, cyclodextrin, has gone from being viewed just as a solubilizer of other medically approved, FDA-approved drugs, to actually being a drug itself, so much so that we're now in a phase 3 clinical trial, and we're anticipating the approval of this drug later this year. So this would be the first. We have no FDA-approved drugs for Niemann-Pick C disease now, but this would be the very first. And that's been made possible, and this whole effort really has been driven by this research collaborative that I told you about. And as part of that, we not only test cyclodextrin and have continued this process, but we've tested dozens and dozens of other drugs, as well as combination therapies, including gene therapy as part of that, all moving forward through that SOR mechanism. So I'll stop with that. Here's some of the organizations that support SOR, and again, I appreciate your attention. I'm sorry if I was a little overboard on some of it. So, thank you. And I am around all day, and if you have questions, or if you want to ask any questions now.
Thanks, Steve. I think if we could do questions at the end, and you'll be here all day, you'll be at lunch. I think two things that I learned from your talk, just to keep it brief. First of all, the complexity of the diseases and being able to understand it is really essential to figuring out what possible treatments, and that sometimes basic research, really tough, but it's really essential to being able to move forward. And the other thing about SOR, really around the collaborations between families, researchers, clinicians, and I think about the Tay-Sachs Gene Therapy Consortium, and really when each of you are in the small groups and we talk about the gene therapy, I think you'll see that progression and the collaboration. So we don't have SOR, we haven't quite done it that way, but that's, but it resonates with me in terms of what we've been able to do
with the consortium model. So that's really helpful to see another example of it. So hopefully people have questions for you later. Thank you very much. Yeah. You want me to, did I use this one? So actually that was a good transition. I'm not going to talk about science. So I could not probably, but I think having Steve talk about the science and all that's involved is really essential. But now it's really important to understand, okay, what does this mean for you? How can I participate? I know that some of you have had experience. You've gone to the NIH or Mass General, you've been here participating in assessments, and you've been working with clinicians to gather data about your diseases. Now we want to talk about something where you can actually do this yourself, enter in data that's going to be really important to moving treatments along. So today, this works. Whoop, that's not my next slide. Okay, that is. Let's see. Okay, so I gotta move that back. Wait, why is this doing this? Okay. Why? Okay. So this is supposed to be the next slide. We're going to be talking about patient insights networks. And are there any of you here who have already enrolled in the GM1 or the Canavan Patient Insights Network? Okay, great. So I want to be here a year from now, and everybody's going to raise their hand, and we're going to see what that's about. And today, we're going to be launching also a Patient Insights Network for GM2. So I'm hoping this is going to get me to the next slide. Okay. So Stacy talked about clinical trial readiness. And so one of the things that's really essential in clinical trial readiness is a registry, collection of standardized information. And in this case, we're going to be talking about certain kind of patient registry. I do want to point out, you'll see in the bottom left-hand corner, it says Invitae. The patient insight networks are developed, run by a company called Invitae. It's a big company. They're getting into genetic testing, all kinds of things. They've helped us set it up. There couldn't be anybody here from the company today to present this. So they prepared some slides. I've adapted them. So I just want to make sure that you had that background. This isn't something that NTSAD or any other patient group is starting. Actually, there's no cost for us, for the patient groups to participate. Also, just want to let you know that. But why is it important? I think primarily the first thing is to form a global patient community. How many of us are out there? Create a community. There's power in numbers. Connect patients to opportunities for clinical trials, research studies. And then also, I think this is a resource for researchers, whether they're in academic institutions or in companies. So the idea is that de-identified data, they don't know who you are, can be used by the researchers to look into how do we develop treatments. So this is a way of helping yourself, but also helping others. So 3 important things about this system. It's online, multilingual. I'll tell you what languages it's translated into already, and it is secure. We'll talk a little bit about security later. There's been a lot in the news about security, so some of you may be wondering about that. So this is not something that's brand new. It was started by a company called Patient Crossroads a number of years ago. There's already 80 patient insight networks that exist for over 400 diseases. There's over 100,000 people around the world from over 85 countries that are participating. And there's one of the factoids that Invitae shared is that you can check off, do you want to be contacted about research? And 88% of these 100,000 plus people said yes. So in our community, actually, there's already 2 pins started, as I mentioned. So Canavan, Illinois started a pin for Canavan disease, and both NTSAD and Canavan Foundation are partners in it. They have to date 100 and this little snapshot shows 114 people entered that have entered into it, and there's a ton of information you can see about it. This is a snapshot of just a few things. So that means 114 people around the world. In addition, Cure GM1 Foundation about 6 months ago started a pin for GM1, and TSCD is also partnering there. I see that's a little bit slower to start up. I think there's maybe 60 people to date. So I think if everybody enters, we can really see, I mean, there should be a lot of people that will be that'll be involved, because there's certainly more people than that, as we know, that are affected by GM1. So thanks to Deanna, who's been working closely with Invitae, we've been talking a lot about the need to set up some kind of what we're calling a patient registry, and to also set up a GM2. And when we started talking about it, we said, well, we need this to be a global registry. So I reached out to Rick, and enthusiastically he said, of course. And then we also, for the Cure Tay-Sachs Foundation, and we've reached out to other foundations around the world, and some of them, they're not formally partnering, but this is really a global thing that even if for some of the European countries, patients have already registered in specific European databases, but you can do this too, and so we can get a global picture, and that's really what we're trying to do. So as I understand it, I haven't registered myself, but it's pretty simple. Actually, I have, thanks to Rick, little half sheets of paper that have all the URLs that we can hand out as people leave so that you can register. A simple registration form, and you can just register, you can finish it later, you don't have to do it all at one time. There are 4 core surveys that are the same for all the PINs for every disease. One about diagnosis, one about medical care, one about general health, one about family history. And then Deanna added one for GM2, social and emotional survey, that we can add, I think, 1 survey a year. And if, let's say, a company reached out to us and said, well, we want to know more about something about GM2, then they could actually, I think they would have to pay to put a survey in, and then Invitae would reach out to everybody that's registered and say, would you like to do this additional survey? One of the other things that I wanted to mention is this is for everybody. So it doesn't matter age. It's for children and adults who are living with disease. If a child has passed away, we still want you to be part of this global community. So everyone here is involved. And also, once you sign up, you'll get to see where are all the people. This is just the US, but there will be a global picture like this showing where everybody is that's affected by the disease. The other thing is that they'll have this view data feature for all these questions that you answer. You'll see what's your answer compared to how other people are answering. So this is, I don't even know what this is about specifically, but it shows certain kinds of alternative therapies, or, you know, what are you doing versus what are other people doing? So you get to see the big picture here. And so, and if you do this, I mean, there's information that could be helpful to you. Maybe some of the surveys, you know, you filled in about health, medical records, things like that, you can download them, take them to your doctor. Over time, you can update it to track changes going on. We already talked about the view data feature to compare yourself to others. I think if you enter in information about physicians, and I haven't seen this myself, but there could be a list of other physicians. And then certainly staying connected is going to be one of the biggest things, is that I think we can put out a notice. Other people, foundations, physicians, industry can put out notices to everybody that's registered in the PIN to let them know about studies, to let them know about meetings, to let them know, I think, really about anything. I mean, I'm sure there are guidelines, but in generally, that's how it's supposed to work. And then again, I think as I started, I think the real benefit here is building a global community. People ask me all the time, how many people are there affected with late onset disease? How many people are affected with Canavan? And we have a sense of it, but we don't really know. And so, you know, there's power in numbers and there's power in your voice of saying, what is this disease like? What's changing over time? How does this affect me? How does this affect my family? This is a chance for you to register. Again, these surveys are all common across diseases. So there's also a chance for them to say, oh, what things do diseases have in common? And certainly it's intended to be a research tool as well. So ideally, and this is really, Invite wrote these words, but they really resonate. I hope this is true. You know, help us lead the way to treatments faster. This could be another tool where you can help build this understanding. So last, about security. You know, this slide is strictly from Invite. So they mentioned that they're leading and providing a secure infrastructure. They're managed according to specific guidelines. And I think the other thing, just to, that researchers only see de-identified information, and that your identity is never shared unless you ask to share it. And you even decide whether or not you want to receive contact information. So the idea is to put you in control. And then you can always, if you just start it, you can opt in, you can opt out. So it provides a lot of flexibility. I don't have any case studies to present to you, but I was thinking if anybody wants that information, I'm sure we could get it from Invite. So, Deanna, who I'm not sure if she's in the room, but you can talk to her because she's set up the GM2 PIN. And then there's a person we've been corresponding with. She didn't give us her specific, Joanne, but I think there's a team at Invite who you can also ask questions of. And I put the 3 URLs for the GM1, GM2, and Canavan Invite. And we don't have it on our homepage yet, but we'll soon have the link on our homepages. And we're not going to, actually, I think what we're going to do is at the registration, we're going to have a computer set up where if you even want to start your registration here, you're welcome to do that. But I hope everybody will want to do this. And we're excited. We think this will be a good complement to what the clinicians are doing and what you're doing in partnership at the academic medical centers and at the hospitals, because that work needs to be done with the clinicians. That's very important. This is complementary. It's not an alternative. So both are important. So next Rick from Cure Tay-Sachs is going to come up, Christy's dad, and he's going to tell you about another new program that we've been looking into that we're going to start that will help us gather more information about use of off-label drugs.
Let me just add to that we have these nifty little yellow sheets that give you the links to be able to sign up for the PIN. So we will have these at the door, and there's specific ones for GM2 and GM1 and for Canavan on here, okay? So would encourage you to register while you're here. And there you go. We'll put them over there, okay? So I am, thanks to, I am Rick, run the Cure Tay-Sachs Foundation. And my daughter is Christy, and she was born in 2006. We lost her a couple of years ago at age 9. So um, I want to talk about something fairly practical. I don't have a lot of slides. I do have a nifty little movie, though, if it works. But I want to talk about something that will be known as the Trend Initiative. And this is something that folks have asked for. So, kind of by show of hands, and I don't know the answer to this, but by show of hands, how many people have heard of this drug Tangenil? There's a lot of hands out there. How many folks have their or have a family member who are currently taking Tangenil? Okay, so there's quite a few. And to add to that, there's quite a few around the world, okay, people that aren't here today. We don't recommend Tangenil. We don't advocate for Tangenil. It's an odd little drug that's been around for a long, long time. And a researcher named Michael Strupp in Munich, Germany, kind of stumbled upon it a couple of years ago. And it's really just available in Europe. It's really indicated for vertigo, and for a bunch of different reasons, it seems to cross the blood-brain barrier. And the anecdotal evidence that we have is that it helps symptoms. It is not a cure, not even close, but it does seem to relieve some symptoms. And we have not had a structured way to measure what that relief is, or what the goodness is, or what the lack of goodness is, okay? And you have asked for that. And my attempts have been somewhat lame in terms of having some conference calls. There is a closed Facebook page that has like 100 members in it now. So there's a lot of interest in Tangenil. So working with NTSAD and with Sue, we wanted to put together a program, if you will, that you can participate in that will allow kind of a broad look, if you will, of what people are experiencing with Tangenil, what results they're seeing, what doses they're using, when did they start it, so that you have a means with which to kind of communicate to each other. What are we seeing with this stuff, and is it helping? Okay. Again, this is an off-label drug. You get it from France. A lot of people are using it, so we might as well talk about it because it's out there, right? Can we roll the video? This is a video from a company called Trend. They do this type of work, and we will soon have a contract with them. This is no charge to you guys. We'll soon have a contract with them, and if we the process with which we will embark on this endeavor is actually pretty simple. Once we have our final contract signed with these guys, and be next couple of weeks, I'm sure, both the NTSAD and the CTSF will provide information on how to become part of the Trend community. So it'll be a series of questionnaires administered by Trend, secure in their database, and it will be somewhat of an interactive database where you can log in and log off and find out what other people are saying. But more importantly, they will put together some summary data, which really kind of looks across the broad spectrum of, I think that's what she's saying, the broad spectrum of what people are seeing out there that are using the drug. And then it will help you decide, is this something you want to use? Is it something you don't want to use? Is it something you want to learn more about? And
what I found is that
all right, let's bring her back to the beginning. Hi, Maria.
Hi everyone, my name is Maria Picconi and I'm the founder and CEO of Trend Community. And like all of you, I am also living with rare disease. My 5-year-old daughter Taya was diagnosed with Prader-Willi syndrome at 10 days old. And after I received her diagnosis, I joined Facebook to find support from others who could understand what I was going through. And what I found is that the reality for a child born today with Prader-Willi syndrome was very different from what the doctors described to us. Patients and caregivers were experimenting with things like special diets, new therapies, trying medications off-label, and some of the things that they were doing really seemed to be working and improving their children's quality of life. As someone who's worked in the industry for nearly 20 years, I was shocked at the enormous gap between what was written in the literature and what was actually happening within our patient community. My husband Chris and I own a digital health technology company, and we've been building software for pharma and hospitals and research organizations for over 10 years. And inspired by our journey with Taya, we decided to build a platform to empower patients and caregivers to share their stories and their real-world experiences in order to collectively evaluate the potential benefit of some of these novel interventions. What's really exciting is that FDA is now looking at these kinds of data to determine unmet needs within patient communities, especially for rare disease. And they're actively exploring how to best incorporate patient experiences into their benefit risk assessments. Everything that we do here at Trend complies with important health regulations such as HIPAA. When you join Trend Community, you do agree to let us share your data, and we use that data in order to accelerate the search for treatments and cures. Names and other personally identifiable information are always removed from our data before we perform our analysis. And our reports only include summaries of community data. For example, these are the symptoms that were discussed most frequently, and these are the treatments that patients and caregivers are using to manage those symptoms. The output cannot be linked back to individual participants, and individual experiences are never shared without your explicit permission. If you'd like to join Trend Community, come to our website, trend.community, and scroll to the bottom of the page and request an invitation. I look forward to meeting you on Trend and working with your community. Thank you.
Great. That was perfect. So in closing here, and I want to get back on schedule because what Stacy's talking about is really pretty exciting. But I take two things from that video. First, Maria, who runs that company, she gets it because she has a kid with a rare disease, and it's one of the reasons that she started all this. And the second thing is that the folks that are on Tangenal, you've asked for a more structured, how do we as a community come together and learn more about this? That's the anticipated outcome of this work, as well as to further research to understand exactly what is Tangenal doing. Should it be part of an ongoing treatment program? And quite frankly, it's kind of exciting to stand here and say we've got another bullet in the gun that we really never had before. And again, off-label use, not something we recommend or advocate, but seems to have positive indications for people that it can help. So with that, I will turn it back over to Stacy. And if you have any questions, hit me up. I'm around. I have the orange shirt on. What's that? Yes.
Because it's not really about Tangenal, it's about Trend.
I know, but at the same time, we talk about Tangenal. I just want to say it's a drug which is available only in France. It's very cheap. You can buy it at any store in France. Yes. And but there is a website where you can buy it from here. If you are interested, come to me.
We do have the website, and it is very inexpensive. So we'll give this to Stacy, and thank you.
Thank you. All right, so this is the last little bit before we break into groups. I know we've heard a lot of really exciting information. I hope that you're all interested in learning more about the pins and about Trend. If you are interested in Tangenal and learning more, and there's obviously a lot of people in the room who will be happy to talk about that later. Can we go do you have the next? Oh, never mind. Sorry, lost my brain for a minute. So the last little bit we're going to talk about, I don't really have as much to say as much as I want to hear if you guys have questions, but we're going to talk about just understanding what the road is to a clinical trial at this point, understanding what we're doing to prepare, and what the researchers are doing to prepare, and what all the things are, the steps that are needed, and who is involved in all of those steps. So to start, we're going to just talk about what a clinical trial is. And a clinical trial really is exactly that. So it's a trial or a study done to evaluate a therapy that's still experimental. And so we've talked about clinical trials for many years. We've talked about how fast we can get there. We've talked about how exciting they'll be. But it's really important to keep in mind that these are still experimental therapies. And so the goals of an initial clinical trial are not just to treat as many people as possible, but we need to first find out, is this treatment or potential treatment safe? And then also, does this potential treatment work? And we always talk, as physicians and as scientists and in this community, we try to keep reminding everyone that safety comes first for a reason, not because safety is what's first and foremost important to make sure that any therapy given to patients is safe. And also that's what the FDA is interested in first as well. So we want to make sure that we're helping with therapies and not harming people. And so again, any initial clinical trial looks at safety first and then at efficacy or whether or not it works afterwards. These absolutely are exciting opportunities, and it's reasonable to be excited and to be hopeful and to look forward to these, but it's important to keep in mind that these are trials. So in our small groups, you'll hear what the researchers are working on and their data showing how the promising steps and the promising results that they're seeing with their therapies. But it is important to remember that just because a therapy looks great in a mouse or in a cat or in a sheep, it's not a guarantee that it's going to help. And that there's, even when this therapy looks safe in those mice and those cats and their sheep, there is still potential that it may cause harm in humans. And so these are all important aspects of trials to keep in mind. It's also important to keep in mind that clinical trials are extremely tightly structured. So when a clinical trial is ready to begin, part of that process includes really developing a specific protocol. That specific protocol includes things called inclusion and exclusion criteria. So any clinical trial, when it's announced, has a list announced of what the criteria are that a patient must meet to be included. And then there are often specific criteria that would exclude patients. So sometimes these are age-based, sometimes they're based on characteristics of the disease or certain things that the researchers are looking for in the disease. But those are really determined by the researchers, by the clinicians who will be doing the medical aspect of the trial, and the FDA and the regulatory agencies that are involved. The protocols also really specify how many patients will be allowed to be enrolled in a trial, and they specify specific protocols and timing. So I think we would all hope that if there was a treatment that went to a trial, that we could treat all of the patients as quickly as possible. But again, with safety in mind, there's also often a protocol that says we treat the first patient on day 1, and then we wait, and we wait, and we make sure that it's safe and that there aren't bad adverse effects seen in that patient before we go on to patient 2. So it's not we treat patient 1 today and patient 2 tomorrow and patient 3 the day after that. So this is all set up in advance. There's also specific signs to look for, sort of slow down signs and stop signs to say, if this were to happen to someone who's treated, then we need to press pause to really evaluate what this outcome was, if it was related to the treatment, and how we can increase the safety if we're going to be able to move forward. And it's really important to remember that these trials are designed partially for safety, but really we need to be able to show an effect. So if we have a therapy or a potential therapy that goes to a clinical trial and it doesn't help, there's no difference in the outcome, then that therapy isn't going to be approved. So for approval, therapies need to be shown to be safe and they need to be shown to have an effect. That's why we talk so much about clinical trial readiness and what those endpoints will be. It's also part of how those inclusion and exclusion criteria are, so that the trial really can help show, hopefully, the beneficial effects of the treatment. So part of the reason why we wanted to talk about this pathway is that there's been a lot of discussion. A couple of people have asked me at the conference already. There's been discussion in the community about expanded access use and how this is or is not different from a clinical trial. Expanded access use in the past was referred to as compassionate use, so you may be familiar with those terms also. So expanded access really is not a clinical trial. It's outside or separate from clinical trials, but it can at times allow some patients access to experimental therapies outside of trials. Sometimes this can be alongside of a trial of a therapy. Sometimes it's when there is not yet a trial for a therapy. For expanded access use, this really requires the researchers, so these are the people developing the therapies, to be on board. There needs to be a healthcare team and medical team in a hospital, or a process on board. And then the FDA is also involved and needs to approve this. So sometimes we get asked, why don't we just do this? Or why not do this now? Or what's the holdup? But it's important to keep in mind, again, that safety is the foremost these things. And so for the FDA to give approval for an expanded access use, they still need to have the same reasonable reassurance that the researchers have done everything possible to make this as safe as possible. So some of the studies that you'll hear about in the small groups talking about things like toxicology studies, which are necessary to show that a potential therapy, we've given really big doses to animals to show if there are toxic effects and when they develop. Those things need to be done for expanded access also before the FDA will allow an experimental therapy to be given to any patient. There also are risks with expanded access. So if a individual is treated through expanded access and there is a poor outcome, if the therapy is harmful, then this risks really shutting down the program. The FDA would look at that data and would say that they would consider this and potentially not allow for a clinical trial. Because expanded access also doesn't use those specific inclusion-exclusion criteria, often is in a limited number of people, it also may not be able to show the type of effect. And it's really important that we have clinical trials with specific patients or numbers of patients to show that there is effective therapies, because that's what's needed for approval. And approval is really the ultimate goal for these therapies, because with approval, then everybody has access to it. Approval also helps us, as healthcare providers, encourage insurance companies and payers to pay the money that these therapies cost, and they're certainly not inexpensive. And so it's, clinical trials are really what is needed for the pathway to approval. And then the last thing that I wanted to talk about with you guys is, and this is a question we get asked a lot because we talk to you about the research a lot. We've talked about clinical trial preparedness, but what is NTSAD's role and the role of similar advocacy groups? So our main job and our main goal is really to bring this information to the families. So to be a communicator of information from researchers and from the regulatory agencies to the families, and to connect the researchers and the families. So as you know, we have many researchers who come to our conferences to talk with you directly about their work. We also have had researchers come to learn from families and to learn about what endpoints are meaningful for families to help them think about the direction that they want to go. And then we've had researchers come and meet with patients and to collect data from patients at our conferences. And so our goal is mostly, and our role is mostly, to be a connector. We also can and have supported much of this work. So we've supported, as we talked about earlier, basic research, and also supported much of what's needed for getting to clinical trials. But NTSAD and similar groups are not involved in the actual design of clinical trials. So we won't have a role in deciding when a research group is ready to submit to the regulatory process. We don't have a role in determining how many patients will be included or what those inclusion or exclusion criteria are. We certainly don't play any role in what the FDA, would request or would require. You guys have been a surprisingly quiet group through this. Yes. I'm sorry. Maybe I didn't make that clear. I sort of anticipated people interrupting to ask questions, but certainly now at this point, if you have questions, please. And Sue has a microphone.
So please use the microphone in the front. Stacy, can I ask you to elaborate on expanded access versus right to try? Because that's a big political issue.
So it's a good question. And right to try has been in the media a lot lately, and you may have heard about this. So the right to try legislation is sort of saying exactly that patients should have the right to try experimental therapies outside of clinical trials. It's, I think, something that is flashy in the media and that legislators like to say, absolutely, people should have the right to try. And I can certainly understand why anybody would want to have that right. It's not always that simple for a lot of reasons. And certainly for a lot of our diseases, when we talk about something like a gene therapy, it's not something where even if you you had the right to try. You can't just pick it up and give yourself gene therapy, right? It's a pretty complex process. And so, I think from a lot of us in the medical community, we think this right to try legislation doesn't have a lot of actual meaning for people, especially in rare diseases. It's also important to keep in mind that, companies are developing and researchers are developing medications and potential therapies, and this is a costly process. It costs researchers in grant money, and eventually when pharmaceutical companies become involved, it's a very costly process to take a medication from the very, very beginning through a clinical trial. And part of and this is part of why we pay medications we pay money for medications afterwards. And so companies often don't have the money, can't afford to make more of that drug to give to patients. So we think about we hear a lot about right to try in terms of things like cancer therapies. If everybody who was potentially eligible for a specific cancer therapy had the right to try it causes some concern. First of all, the company probably can't afford to make that much of it. We don't have a controlled way to find out if this is helping people or if it's harming people. One of the reasons, and I think as families, it's often easy to feel like the FDA is the opponent. But the real reason why we have a regulatory process is that this prevents people like me from saying, I have this treatment. And if you pay me this amount of money, I'll give you this treatment and I will cure you and cure your child. And it's really important that we have proof that therapies work before people try them. And so, unfortunately, I think that the right to try legislature I think it makes our people in government feel good about themselves, but I don't know that it has a lot of true benefit and will really make a meaningful difference for families, especially in rare disease. I don't know. We do have time. There's a question in the back. Okay, we do have time for questions. So.
Hi there. I had a question on your last point. Advocacy groups do not directly design clinical trials or play a role in the regulatory process. So I'm working in a pharmaceutical company, and we like to think that particularly today, and particularly in rare diseases, that we have a patient focus and that we listen to what patient communities have to say in the design of clinical trials. And in another area, so not in this area that the diseases that you're covering, but in a mucopolysaccharidosis area, we went to the regulatory authorities and we were proposing to them a design of a clinical trial that we felt was the right design, which used a natural history as a control group. Okay, so it was a historical natural history. And we all know that the FDA or the regulatory authorities in general will say, yes, well, we do prefer randomized controlled trials. And I think we all understand the difficulties in this field of doing those types of trials. They specifically asked us to go back to the patient community. They said, could you please organize a patient advisory board and talk to them about this design? So I think, things are changing. And there's some examples like that which is really great in bringing the patient community in.
Absolutely. And I think that goes along with some of what Sue and Rick spoke about these trend initiatives and PINs, and that the FDA and the regulatory agencies have really become much more interested in what patients and caregivers think are meaningful. And what I meant specifically by this is that while we can provide information to researchers in terms of what is meaningful to our families and what is important to our families, we won't be the ones saying it's going to be X number of patients, or they have to meet this criteria, or we exclude under those criteria. That's beyond the level of what our direct involvement is. Other questions? Wow. Oh, go ahead.
Wait for the mic. He doesn't need a microphone, but anyway. I just wanted to underscore the point that was just made. I'm not sure who made it. Was it? Okay, hi. And that is one of the dilemmas, I think, that face parents as they consider going into a clinical trial, is that certainly historically there needed to be a control group, and you had to basically say, I will participate whether or not my child is in the control or the experimental group, which is really a heart-wrenching decision. And so you mentioned they're beginning to use natural histories. And I just wanted to underscore the earlier discussion about some of these natural history databases and how important they will be for moving forward so that we can have clinical trials where everyone who is in the trial is treated and they use natural histories as their quote unquote control. And I know they've done it for some of the disease trials, but certainly not yet for these. And so I think that really underscores the importance of participating in all of these registries and helping really work out the natural histories.
And I think this raises something that we haven't talked about as much in this room today, but we've talked about a lot in the past about how one of the challenges in designing clinical trials for very rare diseases is that they're really different from clinical trials for non-rare diseases. So compared to a diabetes drug where we have a standard and you take one medication or the other, or we may not treat anybody. The clinical trial design for rare diseases, it is difficult because of the small size of the population, because the type of intervention is often more invasive. So we're not giving doing control groups in gene therapies where we're injecting, control substances, things that aren't actually medication. And so that can really impact how these trials are designed.
I just had a question because it was mentioned that there's a European registry, or that there are European registries, or are there not?
It's really
loose.
Yeah, the CATS organization based in the UK. I think Dan Louis, who runs it, he enters all the data himself. So it's kind of a database, right? And it complies with European regulations. But I think it's very limited because it's, So PIN is different, but it's, again, complementary, where hopefully we'll gather a global database. At one point we looked into saying, could we use what CATS is using, but it became a little complicated.
And Blythe had a question or comment.
It's actually more of a comment. So I read, all of the things like compassion or expanded access and trials in rare diseases are hitting the news more and more and more as things are accelerating. And as a consumer of that news, and certainly in my involvement with National TASACs, self-interested for these communities, I find it's tempting to generalize. Like, well, if they can do it over on this disease, why aren't they doing it over on that disease? And so I just sort of like a cautionary plug that every path to a trial is unique and every disease is unique. And so read these stories with a bit of skepticism as regards applicability to your own situation, but also I do encourage us to all read them to learn about the trials and tribulations and what's possible and what some of the pitfalls are, because it's just really eye-opening, like the recent story about the MS drug, where the patient population was really pushing for approval and they got it, and now they're discovering that a lot of families are having very bad negative side effects. So it's just good for the eye-openingness to see how complicated this is.
Yeah, and I think it's important to point out that, Stacy was right about the inclusion-exclusion criteria and clinical research trying to come up with protocols, but more and more, and I'm learning this as we go along trying to develop trials, is that we don't even always have the say in inclusion and exclusion criteria. It really becomes a conversation with the FDA about what kinds of things they're willing to accept as evidence, how much change is enough change to be able to allow a drug to be approved. So it has become much more commonly now a conversation with the FDA as to how you design the trial. And when push comes to shove, you have to design a trial in a way that you can get a statistically significant difference between the control group, whether it's a placebo control group or whether it's a natural history control, and the patients that you test in order to get it approved. That's even assuming that it's safe. So it's a long process. We're really learning it's a lot longer process than we thought, and it's much more of a conversation with the FDA. And this sort of idea of inclusion and exclusion is not so much I'm in, you're not in, because an exclusion criteria, for example, could be something as simple as, let's say, and I'm making this up completely, let's say your outcome measure of whether or not you wanted that this was a successful treatment was that you had to show that the amount of brain tissue you had on an MRI scan got more or was less or something like that. Well, if you have a pacemaker or if you have a piece of hardware and you can't have MRI scans, that would exclude you from the trial, not because good enough or couldn't benefit, but because we couldn't use you in order to even see whether or not it made a difference. So when one considers inclusion and exclusion, it's not just, I'm in and you're not, or you're too sick or something like that. Sometimes it really gets down to just, can we use you as a participant to be able to look at an outcome? But it's tricky. The FDA is getting better about working with patients and working with investigators to try to come up with something that is really going to demonstrate an outcome. You have to make sure that it's safe, or at least you think that it's reasonably safe by doing all these studies in mouse models and animal models. Because if the FDA finds, if a patient has a problem early in the trial, like the first or second patient you treat has a problem and they can't tell whether it's because of the drug or just because of something that happened, they put the drug on what's called a clinical hold, and then your trial is done, essentially, and unless you can prove that this bad outcome was not directly a result of the drug. So, you're toasted. So it's a big dance. It really is.
Exactly. And so the goal of this is really to do trials in a thoughtful way so that nobody ends up toasted.
This has been a really rich session here because it's really presented a lot of different ways for us to move forward here, right? And responsible ways. And I think that's a key message. But this isn't perfect science either. And we have to make decisions as families that, are very personal and very emotional too. So I'm going to ask that as we think through this, that with all these tools we have now, on one extreme you have, right to try, which, has its concerns. On the other extreme, you have a very rigid process of clinical trials, and then you have expanded access, and then you have trend. You can begin to see that maybe there's a hybrid model here that's going to get more people treated sooner and have more of us benefit sooner than if we were just doing the traditional phase 1, phase 2, 3 years, whatever it's going to take. And it comes down to where are you and what are your risk tolerances and what's your window of opportunity. So it's a tough question. We don't have answers. I'm not suggesting we go rogue on any of this stuff, but we got some real interesting opportunities right now. So let's go do it.